Hello, everyone. We're glad you've joined us for this live webinar, Sample Management versus Inventory Management. What is the difference? Presented by Kathi, Shea, and Dr. Markus Albertini. I'm Christy Joel of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots and sponsored by Brooks Life Sciences. For more information on our sponsor, please visit brookslifesciences.com. Now let's get started. I would like to encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Simply type them into the Ask a Question box and click the Send button. We will answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing the presentation, click on the Support tab at the top right of the presentation window, or you can report your problem by using the Ask a Question box. This presentation is educational, and it does offer continuing education credits. To obtain your credits, please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. Lastly, at the end of the presentation, you will receive a survey which we kindly ask you to complete. Your answers and suggestions will help improve our webinar program and are greatly appreciated. I would now like to present today's speakers, Kathi, Shea, Biobank Business Lead, Brooks Life Sciences, and Dr. Markus Albertini, General Manager and Director, European Region Services, Brooks Life Sciences. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Kathi and Markus, you may now begin your presentations.
Thank you, Christy, and thank you all for attending today. As Christy said, we're going to focus on inventory versus sample management, what's the difference between them, and why you really need them both for your clinical samples. At Brooks, we're really focused on the sample. Everything we do, all of our products and services are centered around the sample. And that's because samples are really the foundation for how we build information and advance scientific research to deliver new diagnostics and therapeutics so that we can all have healthier and brighter tomorrows. And if you look at the array of products and services that we provide, you'll see that they focus everything from the tubes that the samples are preserved in, automated or outsourced storage solutions, tools for handling those samples. But today we're really going to focus on information about the sample. So you first have to ask yourself the question, why store samples beyond the clinical study? And that's really because they're a key resource for preclinical research and clinical development. Well-organized collections help us get a better understanding of the etiology of disease and the biological pathways that contribute to that disease, as well as allow us to have new therapeutic concepts, new discoveries, whether it be target discovery or biomarker discovery associated with a particular disease. And in the clinical stage, we use this information to help us stratify patients, look at particular subgroups within a population, develop new companion diagnostics, and even identify rare events. And so a good, comprehensive, well-organized sample collection can really make your research and development processes much faster, save costs, save time, and enable collaborations between different institutions. So I want to ask this question, why is it so difficult to keep track of samples? And one of the foundational things that goes into answering this is the wide array of different sample types that there are. So what you see here is a list of very high-level sample types that people have in association with a clinical trial. And within any one of these, you can have lots of variation. So for human samples, for example, you can have blood derivatives, many other matrices that you need to store information on. And the key information associated with each of these samples can vary based on the sample type as well as the disease that's being studied. And you'll find there's a lot of inherent variability within our overarching biobank collections that results in a lot of complexity. First and foremost, most of our biobank collections have samples from many different clinical trials, and those are acquired. We also have material that's acquired from various different suppliers and through lots of different collaborations. Also, within each one of those collections, there can be many different stakeholders, and those different stakeholders often have different purposes. They have different objectives, whether it be for the primary trial or additional research, and also have different areas of the research and development pipeline that they're actually focused on, whether it's in the particular disease's etiology or the mode of action, or doing broad research on the sample sets. Adding to the complexity is the number of locations, both internal and external, where samples can be stored. And the data associated with those samples are also stored in many different locations and many different systems. And so if you ask yourself, do you always know who stores what samples at what place for which purpose, and how are they and the related data stored for your organization? Most of our clients would say the answer to this is no, because there are so many different players involved in storing samples and associated data. So when you're looking for a solution for how best to manage this, really the first question you have to ask yourself is, am I looking for inventory management or sample management? So when we say inventory management, we're really referring to the what, where, why, and how much of a sample do I have. The what is specifically what is that source sample type? Is it, again, is it whole blood? Is it a derivative of that whole blood? What was the source of it? Where is the material stored? As I mentioned previously, you can have lots of different parties involved in a trial, and the samples from a single participant can be spread across multiple locations. Why you have it is really about the intended purpose, and then of course you need to know how much of it you have for that, for your future use. But if we turn to sample management includes all of those things that we just talked about for inventory management, but then brings along with it all of the metadata associated with a sample. So whether that is, patient clinical data, demographic data, family history, consent information, the primary study protocol under which it was collected, all of the methods used during the collection and preservation process, and then I think most importantly, actually the analytical data that was generated from that sample, because as we do additional analyses and we start building data stacks associated with each sample, we're really unlocking the intelligence and getting maximum scientific value out of that, because we can leverage the information that has been previously learned for our future analyses. So what we're looking at now are common sample and data flows for a clinical trial. So it typically starts out with a collection kit going to a clinical site, the clinical site collecting the sample, but also collecting the data associated with that participant who provided that sample. And as the samples and the data begin to move through their workflows, there are many different parties involved in these workflows. On the data side, there are CROs involved. You can have ECRF vendors that are also aggregating data from the clinical sites. You also can have lots of laboratories. Typically, you'll have one central laboratory associated with a single trial, but you can have multiple specialty laboratories. And so within a given trial, you can wind up with multiple different data systems that are storing information, all related back to the sample. And one of the things that we always encourage is proactive reconciliation between those data sources so that while the trial is ongoing, you're resolving any discrepancies between the different data sources. But ultimately, bringing that information together in a centralized system or centralized view into systems is what's really needed for researchers to be able to query that data and find samples that meet their intended research purposes. But this gets more complex when we start looking at biobank collections, because each trial has its own individual workflow and its own individual set of stakeholders and players that participate in that trial. And when we start adding multiple trials together, we can see that we wind up, the picture gets a lot more complex, because now instead of having 4 to 6 vendors associated with a single trial, you're talking dozens of vendors could be involved when we start going across trials, and data often winds up residing with particular study groups and can be very fragmented and hard to get at. And so being able to set up ways in which you can bring that data together from multiple trials, multiple systems, and multiple types of data to create that holistic view is really the holy grail in terms of sample management. So bringing us to the sample management lifecycle, it all really begins with the plan for your study. And as you go through the process of collecting, processing, storing, analyzing your materials, each step of these processes adds additional data and additional information that is important to the downstream analysis associated with that sample, whether it be for the primary study or whether it be for future research purposes. So planning out each step of this process is very critical. And on the left here, you see the normal processes that a sample will go through from the time a donor is enrolled and that initial collection happens all the way down through analysis. When you're planning out your process, really the first questions to ask are what's going to be collected, who's going to be doing the tasks associated with that collection, and when is it planned to occur. The data elements that are going to be collected in association with those processes are also critical. You need consistency and standardization so that information across different locations can be shared. So creating a data plan that outlines the type of data, how that data is going to be QC'd, and how frequently you're going to reconcile the data between different systems becomes instrumental in having good, clean data on the back end. Also, when you optimize logistics, you can also really have a big impact on the overall cost associated with the sample portion of your trial. So looking at the frequency of sample movements that are going to be required and organizing the samples in such a way that really allows you to facilitate access to those samples is key. And so it starts with defining the process and who all of those players are going to be. A lot of people will look at the sample purpose in multiple buckets. What do I plan on using for my active trial? What samples need to be saved for regulatory purposes? And then what will my future use samples be? But in reality, they all really follow the same path. whether they're making, going to a central lab initially or specialty lab, or coming right from the clinic or patient to a biorepository, our overall processes are the same. It's about transporting the samples, registering them into a centralized system, processing, storing, and then analyzing either now or in the future. But really, to accelerate clinical research, you need to make sure that those samples are in a secure environment, that they're audited and handled in a consistent manner to really protect that sample integrity. And I think very importantly, allow for fast retesting or additional testing when needed, either during the active trial itself or later for that future research. And in order to achieve this, you really have to have good transparency into your sample collections. And when you centralize this and you focus on your overall data plan, you can really increase your data quality, your sample quality, and the uniformity of information that you have. And finally, reducing cost. Again, by pre-planning things to optimize your logistics path, look at doing batch processing with possible, and consolidating across vendors can make a significant difference in your overall study budgets. And when considering cost, one of the things that we always like to advise is make sure that you do the math associated with it. So typically people know what material wants to be collected and at which visit it needs to be collected. Then when you start planning your aliquoting plans, you really have to say, what is the total number that I'm going to anticipate? If you're only collecting one sample per patient, then that math is pretty simple. But when you start collecting samples across different matrices and you're looking to have multiple aliquots, those numbers can really increase and drive up the cost of your study. So, and for example, I had one particular trial where it was meant to be a rather modest collection, and essentially by the time that we started looking at all of the single-use vials that were initially requested, those numbers can go up tenfold from what's actually going to be needed for a study. And then looking at your storage format is also important. In the upper right-hand corner, you see a picture of a standard cryo vial. And even in this picture, if you have very good eyesight, you could see that what happens with most standard cryo vials is only a portion of them gets filled. Storing in standard cryo vials, while traditionally that works incredibly well, from a storage density standpoint, they actually do take up a lot more space. And so planning your storage in a way that you can maximize the footprint will help save storage costs for you in the long run. And with that, I'm going to turn it over to Markus.
Okay, thank you so much, Kathi. Yes. So I call this slide Preparation Supports Sample Management, Management Supports Preparation. And that's heading in the direction that we are looking for samples that are really best fit for our purposes. And especially if we want to use the samples for broader research later on, want to store them for a long time, it's really important to focus on certain aspects that may help us to generate the quality, to have the purpose of use and the scope of use in place we need later on, maybe many years later on, and to stay compliant with our sample and sample usage. So purpose and scope of use, that is very important because we need to take this forward into the ICF working, and we have to align this with regulatory or IRB requirements. Meaning what we state in the ICF is written there, and we have to comply to this. You have to use the sample and, of course, also the data. I put data several times here on the slide because without the data, the sample is not that useful as with them. We kind of have to stuck to this, and of course, we may also have to go back to the ICF to dig out relevant information. What can we do with this sample? Then, of course, we should have a clear picture about the sample types and the taking procedures, known how we did it, what kind of sample we have, and maybe also what kind of sample we would like to have in our collection, like kind of streamlining this. Sample logistics, that was already addressed also with considering cost. It's very important to route the sample, have an idea. Do we want to route it to the central lab or maybe directly route it into the biobank? Do we want to move it into the biobank after reconciliation, or can we consider to do it before and then tackle if something comes up during reconciliation later on, and then have a clear picture how we want to store on the geographical side and what are the storage conditions for our sample collection. Okay, then at a later point, of course, we also need to access our samples and having a good sample inventory in place so that our researchers and every other stakeholder can really see what we have there to come up with ideas to do their research planning is another important building block in the concept of being really prepared for setting up a good sample collection, a long-term sample collection. In the end, all this together enables us a sample analysis as we wish or foresee it. Yeah, easy to select the samples, having the right samples in place, and of course being allowed to use the samples for the purposes we would like to use them. Looking a little bit deeper into the purpose and the scope of use, I try to summarize this here in this overview scheme, starting with mandatory and ending here with voluntary. Mandatory means how can we include our purposes and the scope of use into the informed consent for the patient. What will the ethic committees accept? What is regulatory possible? And also data protection has to be considered because also there it says we should inform the patient or the donor of the sample, what we plan to do with it, and of course also with the data. And then if you look here, if you just go for the indication, yeah, the classical or traditional clinical trial, yeah, looks into one indication, a disease, and we try to show that our compound we bring forward in that trial is safe and it is efficient. But this is very often not broad enough. Yeah, basically it's the mode of action we are looking after, and it's a class of compound. So taking forward the front-runner compound, and the front-runner compound shows some problems, some issues regarding safety or efficacy, we have to go to the backup compound, meaning our ICF may state something that does not allow us to use the data for research on the backup compound. Going for the class of compounds or the mode of action helps us to overcome this limitation. We may even jump into another therapeutic area if the mode of action and maybe even the compound that was originally developed for an oncology therapy is also very helpful, like in respiratory diseases. So there's some examples out in the world, yeah, where that happened, and we would like, of course, to use our samples and data also in that case that we originally collected in trials for oncology. And then, of course, you may look into biomarkers, biomarker research, and that could be added as an additional objective. So then you see the lines in the middle, to the left or to the right, basically defining a scope where you may have a good chance to take your ideas, your purpose and your scope of use forward in the main consent as a mandatory requirement for participating in the study. The topics below the line, there it becomes more difficult, and it may you may need more efforts to discuss with your ethic responsible persons if that should be done, if that will be allowed by the ethics committees. That means, for example, going for the therapeutic area, yeah, still closer to the indication, than asking for use of the sample in all biomedical research. And then, of course, to going to the right, yeah, would be future research. There you also have the time span. Will they accept storage of 15 years or 30 years? What do I need for a storage? And depending on that, it may be mandatory or more voluntary. That means the patient or the donor of the sample becomes the opportunity to freely decide, I want to provide my sample for future research, or I do not want to do that. So we made some experience that depending on the indication, if it's voluntary, you will receive, like in diabetes, about 60 to 70% of the patients that will agree to provide their samples, up to 90% if you go for oncology. Like the more sick the people feel, the more open they are for providing sample and data for future research. So taking this into account, how could a biobank or a repository look? Yeah? If you see to the left, it says clinical trial samples. That's basically the light blue block. Yeah? This is the sample collection where we would have 100% of our study participants, samples of all of these. And then you combine study 1, 2, 3, 4, 5, many studies. Yeah, and you can always go back for your original purposes to this collection, and you will, in any case, have 100% of study patients to answer your question. We call that here the clinical development biobank, yeah, and for the definition mode of action, giving you freedom to operate, but limit you if you want to go around about research related to the mode of action or the class of compounds. On top of that, you could, for example, ask them to also voluntarily agree to further research, to broader research. Yeah, basically, you could then use a subset of your samples, 60 to 90%, also for open research questions that may come up over time. Then defined as a research biobank, basically the dark blue box and the two gray boxes, that would be a sample collection enriched also for samples acquired from vendors. Yeah, because in a clinical trial, the more invasive you go, the more difficult it is to get the samples. You may face a need to have also additional samples in your collection. And then, of course, collaborations. There are many good, undertaken biobanks out there in the world, and clinical centers may also help to further enrich your biobank in the end. So how can we best reach that without being too complicated, having to do too exhausting reviews of our ICFs and kind of to keep control over all of our samples and studies, and they come from different places, are stored at different places, maybe as Kathi lined out before. So my slogan would be, keep it simple. Work with standards and stick to them. Yeah? You will have a good understanding about what you have in your biobank and what you can do with it while staying compliant. And if I talk about standards, it's pretty simple standards what I propose here. Meaning I have, before I start, I have an idea what are the minimum studies in my organization in which I will perform biobanking or collect samples for biobank. Like for a pharma company, it could be all pivotal studies, all studies that help to bring a compound to the market. Or what is my maximum long-term storage timeline? For example, 15 years. Or maybe you say 30 years and take the 15 years as a fallback position if the regulatories or the IRBs do not agree to the 30 years. Then, of course, have a good understanding what sample types and matrices I would like to have per therapeutic area. That helps you in the end if you want to use your samples across trials. What do I need? How should my urine be collected? Do I need a sediment if I collect urine of the urine? Yeah, that may depend from therapeutic area to therapeutic area, and have a good understanding what I may need in the future helps you to have it in place. Yeah, to collect the right thing for the future. Then, of course, do I want to have leftovers? With leftovers, I mean not a backup sample that didn't have an additional resource cycle, but a sample that comes back like from a analytical laboratory, yeah, like in oncology, where we shouldn't take that much blood from the patient. It may be a consideration worth. In other indications, we may say, no, we don't want to have that in our collection because we are not sure about the quality. And then, of course, as Kathi already touched before, aliquoting. Yeah, when and where do I want to do the aliquoting? At a study site where the sample is taken, at a central lab, or before it goes in the biobank, or maybe even when I take it out of the biobank. Yeah, it's the important thing is how many resource cycles do I have in the end. Then, of course, sample taking procedures and the storage locations and the minimum scope of use, as explained before. Do I need 100% of the patients, or can I pay a price for doing broader research by not having 100% of the patients? One last thing, ICF and CTP wording. There should be good templates. This should be pretty stable. The wording shouldn't be changed within a trial or maybe even above trials. So you can be pretty sure it's the right thing. It's the right message in the ICF. And basically, I only need a yes or no. The ICF is there. I know what's in there. If you can establish that in your organization. And ICF and CTP, we found very often that the ICF wording is in conflict with CTP wording and vice versa, and that may generate a problem. So we recommend keep an eye on this. Then linking clinical and biomarker data, we believe this is essential to link them to the sample, to have the full data, the full data repository available for good research. To really have the view, what data do I have available, and to have the opportunity to get this data for my analysis. Also that needs to be established by the ICF. And then, of course, and that sometimes generates additional issues, I do research with my sample, and that research very often doesn't go back into original, like in the clinical trial database or wherever your data are stored, but they are more or less free-floating and with the researchers that performed the research. And that may give you relevant additional information about your sample. You want to also include this into your data collection that is tagged to the sample. And all this given data privacy and protection, especially now with the GDPR in place in Europe, is a very important aspect of all our future analysis to keep the privacy of the patients or the donors, and we should do that with providing their sample and their data for research. They support us, and that should be well acknowledged. So what could be safeguards to protect the identity and the privacy of our sample donors? Of course, we only see, or most of us only see, the coded sample or the coded data. It comes with a pseudonym, double-blinded, single-blinded, depending on how we want to do it, what we want to do with the sample. We may decide to anonymize it at a later point. All this defines the measures we have to take to keep the privacy safe of the sample donor. Important is that samples and also the corresponding data are stored in a secured and access-controlled environment. That means we need to know who has access, and we need to know what the people that have access with the data. And of course, we should have a process in place where we have a committee where we ensure that all the right steps are taken that control and allow in the end sample access and use according to the ICF. Then standard operation procedures, at least in pharma, they are standard. We need them. We work under GCP, and also the samples are collected under GCP, and they stay under GCP. Also, if we use them later on for research purposes. Of course, we do not validate or do not need to validate our equipment and our methods for research purposes, but we still need to have the audit trail on this sample. Okay, and in the end, of course, we should also destroy or use the samples according to the informed consent, meaning we need to have the timestamp on it and we need to know when they have to be destroyed. I just made a little box down there to the right addressing the GDPR, summarizing few of the key elements, like the data donor has the right to be forgotten. He can request that his data can be deleted. That's not that relevant in the context of GCP, because there we have a law that makes us keep the data for a long time. Yeah, but if we go into research, it's more critical. Are we allowed to keep the data? The data deletion may compromise our research results, or do we have to delete the data? Questions that are really discussed between data protection lawyers, between scientists, yeah, and they're still not all very clear. Yeah, then there's the right to know who uses the data, and the data donor can request information about what we do with with this data. And most importantly, before analyzing any data, before processing them, we need a legal basis to do so. Otherwise, we wouldn't be allowed. The legal basis could, for example, be the ICF. So, what are the challenges of consent management? Of course, we have a diverse, if you go for a global trial or study, ethical environment where the regulations continually evolving, new ones evolving, the anticipation of regulations change, and they may differ between country to country, yeah, because of cultural heredity, and that makes it, of course, pretty difficult to work on a global basis with one ICF and to fulfill all the requirements from the ethical perspective. So going a little bit more in detail, it's of course we need to ensure the identifiability of the sample. At the same time, we need to ensure that the donor is not identified. We need a clear view on the breadth of the informed consent, the authorizations we have in place, and the requirements we have to put in place to keep control of this. Then withdrawal of consent, we need to take care of this, and the longer the sample is, the more difficult this is. Yeah, like receiving a withdrawal after 15 years in the organization, so that doesn't happen that often, but that may provide us with considerable challenges, depending how we are organized. Privacy restrictions I already touched, genetic research information. We need to keep track of this. Yeah, are we allowed to do whole genome sequencing? Do we want to do whole genome sequencing? Do we ask the patient for that? Did he agree on whole genome sequencing? Then storage and retention of samples. How long? When do we have to discard? For what can we use it? And transfer of samples, of course, are we allowed to or export the samples? And the same is true for the data. Yeah, are we allowed to export it to the US, for example? What does data protection regulations in Europe say about that? Intellectual property, ownership, and commercial use is another aspect, and of course, return of research results. What do we have to return to the patient, to the sample donor? Having this all said, I highlighted legacy studies here, because legacy studies, they require most of the time manual review, and they are the studies that really hurt us, because there we have to do lots of footwork to really have the right picture what we are allowed to do with the sample. So hopefully e-consenting and better data integration will help here in the future. Taking all this together, basically these are the factors that define the space of our sample usage. So sample access model, basically we need to annotate the sample pre-analytical variables, and we need to have a clear picture, develop clear reports for reviewing groups, meaning we group all the samples studies there according to the usage, what is allowed, yeah. Set a threshold for level of review required. Like a legacy sample, we need much more review efforts than we would need for our studies where we had our standards in place and where we can be sure that the standards were followed globally, yeah, from country to country, that the ICF didn't change from the wording and from the meaning. So thereby we can group into low impact samples or studies, meaning only minimal review is required, or call it a risk-based approach. We can be pretty sure that we stay compliant when we use the sample for a certain purpose. And the other one, of course, is high impact, where we need a stringent review, where we have to go into detail, where we have to really carefully look into our documentation in our data to get a full picture and to be really on the safe side when we take the sample out of the repository. So overall, there are 1,000 laws, regulations, and guidelines on human subject research in place. And of course, we are talking about more than 120 countries where these regulations apply. And the regulations, as said, there's a geographical variance, there's differences. It's not in each and every country the same. And therefore, it's really important to keep oversight. In principle, it can be said that 8 categories that are covered by such regulations and guidelines. In general, those applicable to most or all types of human subjects research. So that's really to our heart. Then, of course, drugs and devices and the development and approval, clinical trial registries, research injury, privacy data protection, also very important for us, human biological materials, genetic research, embryos, stem cells, and cloning. So if you would like to get a better overview or you look for a special country or so, you may use this web link. It's a very nice tool and it's really worth visiting it if you didn't do already so. Lots of information and very well done, brought together. So having collected samples, having them in a systematic way in a biorepository, in a biobank, of course, the collection will grow. It will grow over time more and more and more. And we should also have a strategy in place how we assess this collection. What do we really need? Where do we have quite an overflow of samples? Can we use the sample, yeah, to keep it really to the purpose, your collection, yeah, also your research strategy may change. And for that reason, it makes totally sense to assess the collection. It may save you money in the end, and it helps to keep you to keep overview. So starting with the clinical data, of course, the first question is what clinical data do I have available? Then is the data I have available of importance for other studies that may be conducted? So the scientific impact they may bring. Can I do cross-analysis with this data? Will they help me in various research projects? And then, of course, the data quality. Is the data trustworthy? Do I have control parameters in place that can give me this information? Depending on what your outcome on this question is, you say no, and then you may discard the samples because the databases is not good, and the information the readout of the sample will give you may only be limited or worthless. The second question, if you have this answered with a yes, will be the consent, the ICF again. Yeah, will it allow me a broader use or use according to what I plan to do? Yeah, do the samples and the purposes meet the definition of use subject research? Has the ICF been reviewed for use broader than the original study plan use? Does the ICF contain a provision for use beyond the original purpose and collection? And then, of course, time, genetic use, disease area, specific consent, coding requirements. This is very important, and that may also result in a yes or a no. So we have all this in place. The sample seems to be quite usable. We should keep it. Yeah, that means after a final suitability assessment, yeah, what is the potential use of the sample starter? Is it fit for the purpose? We may have to take the decision, yes, it is, and then we keep it. But going through this scheme, asking this question, this helps you to really build and maintain a biorepository that suits your purposes, that has the quality you need, and that has also the amounts you will you foresee and you can afford for your future activity. So, summarizing this, form, structure, function for sustaining your biobank. Basically, this means define and annotate the collection, the primary study, what are my defined ancillaries, yeah, my aliquots, what follows from the mother sample, what are the future research purposes and what I'm allowed to do for future research. Then it would be good to set a threshold. Do I have to expand my collection? Do I have renewable resources? How to bring this renewable resources when needed? And of course, it could always be considered to also acquire limited resources that I cannot get, for example, through my clinical trials. And I think that's also very often something happening in organization, that acquired samples are not in the same system than the collected samples are, meaning they are not seen. Yeah, and very often researchers do not really know what kind of acquired systems I have in my organization. Yeah, and also bringing this into one samples inventory may considerably help to enforce and enable research. And then, of course, culling of abundant resources may also be considered. Overall, this means to develop an impact-based review process, yeah, of the collection. We are doing the collection assessment, and of course, taking also into consideration the individual research questions I may need this collection for. So, what, in summary, are the critical factors for successful inventory and sample management? Starting on the left side, at the bottom, sample and data standards. It's sample and data standards. Do I have the right samples? Are they collected the right way? Do they have the quality I want to have? Is everything in place? But it may also be the standards how I collect the samples and data and how I'm prepared to collect them in a way that I can use them later on. Then single view of inventory, a very important aspect. If you want to plan your research, your experiments, you need to know what you have to work on. meaning having a view on the relevant data that gives you the information what may be the samples that are valuable for your plant experiments, give you the full information if you have sufficient numbers of samples, if you can do the experiment, and how strong your power will be for that experiment. Then, of course, consolidate sample assets, bring all the assets you have together, make them visible in one inventory tool, have the full overview what you have. Then linking clinical and biomarker data and sample information together also is the basis for your research, for the quality of your research. And then repository assessment, as said before, helps you to keep focused, helps you to be the best, to undertake the best for your purpose sample collection. Taken together, that will help you to have a better management maybe in place for your biobank. So, comprehensive data visibility, better utilization of samples, faster sample access, better data quality and uniformity, regulatory compliance, reduction of costs. Of course, that may be what you gain from a pragmatic sample management approach. So, coming to an end, we all know someone that is impacted by disease. So, the samples are so precious to us because they are the key to unlocking scientific discoveries. The proper collection and preservation of our samples and their associated data will allow us to advance science to create healthier and brighter tomorrow for all of us. Having a good sample collection in place, making this available for scientific research, will support development of new therapies, compounds, and in the end will be a good part on the way or the basis really to make a change regarding diseases. So, having this said, we are open for your questions.
Thank you, Kathi and Markus, for your presentations on how we can know the difference of sample management versus inventory management. So let's get started with our Q&A. Now, just a quick reminder to our audience on how to submit questions. If you haven't already done so, just click on the Ask a Question box located on the far left of your screen and click the Send button. And as a reminder, any questions we are unable to get to today, Kathi and Markus will be answering them via email, so don't worry if we don't get to your question right away. Okay, our first question. Now, Markus, this may be for you, I believe it is standardization of sample collections achievable?
Yes, it's achievable. So I was running biobanking operations globally for quite a while. And the important thing is that you have the stakeholders in your organization in the boat, meaning you have to do training, you have to convince them to support this. And if your organization supports this and it is watertight, it holds through, it works. And that helps a lot. It facilitates the use of the sample collection, basically every step on the project a lot. And in the end, you may really be able to reduce the question, can I use the sample for that purpose, to yes or no? Do I have an ICF in place or do I not have it in place?
Thank you for that. Now, Kathi, let's come over to you. What policies do companies have in place for return of research results and or incidental findings?
Well, Christy, I think that, we actually find that this is a question that lots of different companies struggle with. Research results are just that. They're still research. The biomarkers that are being studied have not yet been validated biomarkers for that particular disease, and we find that most companies findings that they have that are on biomarkers that are still in the research phase they don't release to the study participants because it's not known whether or not that's a true result at the point where we're still in the research mode. Now, when there's an incidental finding associated with a validated biomarker, that's quite different, and that typically does get returned to the clinical site for reporting to the patient.
Thank you, Kathi. Now, Markus, let's come back to you. How frequently do IRBs agree or not agree with having future research sample collections mandatory for participation in clinical trials?
So my experience is, maybe others made other experience, having future research that goes beyond the indication or the disease of the original clinical trial, having this mandatory in the agreed by the ethic boards is very, very difficult. Yeah, meaning the moment you cannot inform the patient, and I think that's the critical point about what will be done with your sample, makes the ethic experts say it should be voluntary. You cannot tell the person in the what the sample will be used for. You should give him the opportunity to say yes or no. I think it's fair. It doesn't make our life easier. We have to live with the situation and have to find good ways that we can do good research. The research is also for the patient and the donor in the end, yeah. And at the same time, acknowledge the rights and the needs of the sample donors.
Thank you, Markus. Now I'm going to pose this next question to both of you, and feel free to weigh in. My question is related to the challenges of consent management. Now, when a patient is no longer alive, are the samples destroyed, or are they still accessible for what they consented? Are there any specific rules or regulations in place?
Why don't I take the first stab at this? So typically samples are retained after a patient dies. If they have consented for their samples to be used for research purposes, then the samples and the data that goes along with them typically are preserved. One of the interesting things is in the United States, once a person is deceased, no longer fall, their samples and data no longer fall under the human research protection laws because they're not considered human research anymore because they are no longer alive. But I'll tell you that most groups that I work with, really all groups that I work with, actually treat samples and data from deceased donors in the same manner that they do from live donors, meaning that they respect the individual permissions and restrictions of use of that sample even after a person dies. Markus, do you have anything to add to that?
No, I fully agree. I have the same view, and I think the only thing that may to be considered is if we go for genetic analysis, then of course there's also information about the family in there, and I think that may be a point of discussion, where we do not have the full picture right now.
Thank you, Kathi and Markus, on that. Now, we have time for one last question. Kathi, we'll close with you. Does Brooks offer a sample management system that can track all the variables discussed?
We do. We actually have inventory management systems and sample management systems available. Our inventory management system is Freezer Pro, and our sample management solution is Biobank Pro. And actually on the screen right now, you see a link to our website. You follow that link, you can get more information on those systems.
Thank you, Kathi and Markus, for your time today and for your excellent presentation. I would also like to thank LabRoots and our sponsor, Brooks Life Sciences, for underwriting today's educational web seminar. Now, before we go, I would like to thank all of you for joining us. And as a reminder, those questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. Now, a survey will pop up on your screen, and your participation is greatly appreciated. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. You will now be redirected to the registration page for our upcoming webinar on August 14th at 9 a.m. Pacific Time, Continuous Improvement in Sample Management, presented by Bill Levitt, Jay Patel, and Zach Kogler. We hope to see you there. Bye-bye.